Hello friends, welcome to the part 2 of lecture 1 of week 9. So in the previous part we discussed two important methods of function annotation, that is BLAST and AGNOGs, and we will continue with some other methods which are available and widely used. So gene ontology is one of the common methods for large scale function annotation. Basically it does for single protein, but you can use these servers to do large scale function annotation. So basically what is gene ontology? Function, as we understand, there can be a molecular function which is happening at the how the what is the role of that protein at the molecular level, okay, what part like catalysis or what activity it is doing. Then the cellular component, that is the location where it is doing that job, okay, and the biological process, that the larger picture where what role it is playing in that biological system. Okay, so taken together these three basically amounts to a set of comprehensive picture of what a protein does in a system, right? And so basically for each protein you'll get three IDs. One is molecular function, cellular component, and biological process, and based on that you can do the your analysis, further analysis. This is the gene ontology resource. Okay, so it is available at geneontology.org, and it's a very comprehensive resource. So AGNOGs and KOG are two parallel, basically one approach, and gene ontology is another parallel approach for assigning function to a protein. Okay, for example, it is for one query it has given function into the biological processes, molecular function, and the cellular components. Okay, so basically cellular component means where in the cell it is belonging to. Okay, molecular function, like what activity it does in the cell, like transferase or oxidoreductase or ion binding, and the biological process, like it could be metabolic or cellular or response to stimuli, signal transduction, etc. Okay, and like similar to AGNOGs, there is, or you can say the ancestor of AGNOG is KOGs, okay, which is a cluster of orthologous group which is still available at NCBI, and the first version was created sometime in 1997, and then it was later updated. But the same concept and with the classification that was used at that time, okay, most recently in 2014, is currently used to develop other tools such as AGNOG Mapper and AGNOG database. Okay, so basically it defines the proteins into various functional categories like these. So for example, I have 10 proteins, so if I do search against the KOG protein database, so it will assign the protein into any of these categories. So it can be assigned as a function, for which means like RNA processing modification. So basically that is information class, okay, that includes translation, transcription, replication, okay, or basically one of these A to Z categories, and each category has a single letter code and like what it the expanded definition of that function. Okay, so basically your proteins will be assigned in this, and basically then you can do the functional pie chart or the bar chart analysis, that which is how much is what. For example, you can see that okay, in a cell, like for a genome, basically a lot of functions were unknown. Most abundant functions were signal transduction and post translation modifications, okay, and very rare functions were, or like fewer functions of cellular structure, defense. And basically you have to see these in the total picture, that sometimes it will be the case that those kind of functions are always more abundant in a cell. Okay, so you have to compare with the expected proportion versus the observed proportion of the cellular the functional composition, and then see that what is overrepresented or underrepresented. Okay, another database for functional annotation pathway mapping is the KEGG database, which is a very comprehensive database that classifies a protein into various KEGG. KO numbers are assigned to proteins, and then those KO numbers are linked to the pathways, okay, molecular pathways, metabolic pathways. So KEGG is one of the important resource, and it provides a server called KAAS, KEGG Automatic Annotation Server. So where you can upload your data set and it can classify those into various KOIDs, pathways. So this is available at genome.jp/KEGG/KAAS, and so basically there is a database which they have made for reference proteins assigned to various pathways, and you have a query. So your query will be mapped against those reference proteins, and based on the closest match, basically that is done by local alignment by BLAST, okay, or by ghost S or ghost Z. So that will find the closest match, closest KOID, and that KOID is then linked to various pathways, which is like pre-assigned in this case. So in the result, you will get the information about the function as well as which pathway it belongs to and what reaction or what role it does in that particular pathway. So you can paste the data here like earlier or upload your query. So these are the common options with most of the programs, that you can either, for small searches, you can paste some text in a standard format such as FASTA, or you can upload your file containing multiple sequences and get the result, okay, like this. So basically you can define your selected organisms and the query you have defined here, okay, the method that you want to do, bidirectional hit or single directional hit, okay, and basically it will give you the output that, okay, what all pathways were found, like in glycolysis, which is the map ID is 00010, 34 proteins were assigned, and okay, like that okay. So both the bidirectional and single directional hits, right? So this way you can these can also be translated into the pathway maps, okay, which shows that in a reference pathway which all enzymes are highlighted. That means those are present or found in your system. Okay, so like this is again the output example. Like you can see that in the ribosome and these pathways, polyferin metabolism, or you can see pyruvate metabolism, right, RNA degradation. So various pathways and number of proteins mapped to those pathways. Then okay, so these are based on alignment and where BLAST and other methods are performing, okay, even KOG or eggnog or KEGG that you use, okay, are also using BLAST to search against their databases and giving you the hits, okay, and then further annotation. But sometimes your these methods may fail because BLAST does not retrieve you anything, okay, then, or because basically our knowledge base is restricted by whatever we know or the proteins that have been observed in the past. But okay, but there could be some novel proteins for which nothing or no close hit is available. So those can be assigned by other methods such as pattern matching, Motifs, fingerprints, right? So these are some of the databases like PROSITE, Pfam, prints, COGs, blocks, which can do based on partial mapping or some domain or some small motif which is present, right, which can be used to tell you some clues about, okay, like for example ATP binding domain is present, that means at least this function will be doing this kind of job. That can also tell you its cellular location and other things, okay. So this is the PROSITE database. Basically you can see that so it's a database of domains, families, functional sites, patterns which are associated with proteins. So most of the proteins can be assigned to various protein families based on the domains, and similarly every set of proteins or family of proteins also have some conserved patterns or motifs which can be used to actually connect or basically associate that protein with that functional category. Okay, so basically you can enter your sequence here, and like you can see that these are the various domains which are present in a protein, okay, right. So basically this way you can search, and this will give you information about which part of the protein contain that active site or domain or right, or pre-annotated sites which were already known. So even if you don't know the function of the entire protein because it is not known or no experimentally validated yet, you can get some clues about its function. Okay, so basically idea is that most of the proteins can be grouped into some families based on like the similarities between them, and there are always some critical regions in a protein which are better conserved because they are important for the three-dimensional structure or folding or active site or binding to various other components in the cell. Okay, so basically those cannot change, and those conserved sites or less prone to change sites are important to determine the function of a protein. Okay, so basically those are can be used for the function annotation. Okay, then just to give you an idea about like what a pattern of motif is or motif is, these are short stretches in a protein which can tell you a lot about. So basically those will be commonly present across proteins, okay, of that particular function, but because those proteins or could be in different genomes which have followed different rates of evolution, and also may have undergone various changes due to the evolution, various residues in a amino acid, and okay, based on species specific changes or adaptations. So it is, instead of looking for a like conserve or basically identical motif, that is for example I have a sequence of amino acids which is 12 amino acids. So instead of looking for exact 12 amino acids, I should be able to allow some match mismatch at those 12 positions, so that I can get I can cover the diversity of that particular functional motif without changing its actual function. So that is why a good signature pattern should be as short as possible. It should be able to detect most of the sequences which are associated with that protein function, okay, and should not give too many false positives, and should exhibit high sensitivity. That means it should be able to find more and more with high specificity. That means whatever I am finding should be correct also. So like this is one example query, like tyrosine protein kinase FASTA format. Okay, so it gave a motif such as basically you can see that LIVG, okay, P. So basically this tells that among these, any of these can come at this position, any of these can come at this position, right? Then X means that it can be replaced by any amino acid. So basically motifs we also did in our earlier classes, right? So this tells that can we deduce a protein or a functional component or a part of that protein subset, okay, into a motif which can generally represent a class of proteins that will that should be containing that motif because they are involved in that kind of function. Okay, so like in this square bracket you can see that this is position number 1 and FILV. So in among these 4, any one can come here, okay, followed by Q, then 3 any amino acids XXX, then among R and K only one of those can come, followed by G and any 3 amino acids, and like that. So this curly bracket means that anything can come but not this, okay, not S or T. So this way you can say that for each position in a motif I can define that what all amino acids can come, what are allowed but what are definitely not allowed, what should be the gap between two conserved sites or residues in a motif, okay, like XXX and like that. So this way I can deduce a identical okay, fingerprint or okay, that I found in a protein to a generalized motif which can be able to find any or more of such proteins by looking into this kind of motif configuration. Then another way of classification is based on protein families. Okay, so basically most of the proteins can be assigned different families, and this job has been done very well, and basically this data is also used for alternate classification based on hidden Markov models. HMM based Pfam profiles can be made, which can be used for, instead of doing a blast you can use those also for searching proteins. Okay, So it's a very comprehensive database, contains Pfam A and Pfam B database, okay, which can be used for both machine learning based analysis and also direct protein annotation. So okay, so basically as I mentioned, like Pfam A and Pfam B are the two databases, and generally like this Pfam A is used, okay, and Pfam B is the database of small families, right? And so how to search it these days? It is like there is InterPro, or you can say Interproscan is a software which is a comprehensive software to search multiple databases, such as either it could be Pfam, or it could be the PROSITE, or right. So basically profile. So you can see that it has signatures from profile, HMMs, patterns, composition prediction, okay, domains, families, features and sites, right, and CATH database, Pfam database, smart database, okay, and HAMAP database, PROSITE families, so prints. So a combination of all these are together in InterPro. So if I search in any of these, right, I may get partial hits, but if I search directly into this, I will get a comprehensive picture of which all information I can get from all of these together. Okay, so this will give me an alternate way of assigning function to a protein rather than BLAST or AGNOGs or KOGs. And like in terms of what it contains, like about 3000 superfamilies, more than 21,000 families, 13,000 domains, right, and information about various active sites, binding sites, conserved sites. So it's a very comprehensive search. It will be slow. For example, this is the search page for InterPro, okay, and you can enter your query here, or upload your query, right, and like this is one of the query that is a FASTA format, and it can be uploaded. So it detected that is 532 amino acids, and status is still not appeared because still going on, right, and this is your job ID, which you can copy and you can use it later also for pasting it here. And because Interproscan will be slow, okay, so it is not that you will get your results instantaneously. So it may take 30 minutes to 1 hour, or like even more, depending upon your query size. So you should copy this and keep it safe, and later on you can retrieve your results from there. You can also download Interproscan locally, but you should have sufficient hardware support, good system, so that you can download it, as well as along with the different databases that you want to search against. Okay, so this is also available for standalone download. And in addition to these, basically there are several other databases which are covered in Interproscan, but you can also search independently, like PROSITE, Prints, PDB, Blog, Smart. So all these like will give you prodom, okay, protein domain. So will give you partial information about the function of a protein, which may not be the like final function of protein, but at least partially, that at least this protein of a novel type can do this kind of job in a cell. Okay, so basically in this lecture we learned about the methods of protein annotation in addition to BLAST and AGNOGs, which we did in the last class, and these will help you to comprehensively assign function to a single protein or large data sets, which you will derive from genomic or metagenomic analysis of either could be proteins or ORFs, okay, and you can assign function to those, and then later on those can be, and basically not only functions, many times like KEGG or AGNOGs will also give you a comprehensive picture of what role they are performing and which pathways they are belonging to, and then it is based on your interpretation. You can find clues for like based on that analysis, that what could be the like functional complexity of an organism, and some specific proteins, and based on their abundance. So that is like second level of subjective analysis, and so this is good for today, and we'll meet in the next class. Thank you.